Hi guys, welcome to chapter 3, part C. We're actually going to break this section of the chapter down into two parts just to keep the videos a little shorter. A lot going on in this section. We've got something called mitosis and something called meiosis. They both involve cell division. This video will strictly talk about mitosis, okay? All right, so let's just talk about cell division as a whole. In order to grow and in order to replace old dead cells, your cells need to divide, okay? And the cell cycle is divided into three main steps. We've got what's known as interphase, mitosis, and cytokinesis. A lot of times the term mitosis is used to talk about all of this, just cell division is mitosis. But really the interphase and the cytokinesis are kind of bookends for mitosis. So we will separate it out a little bit. You could see in this diagram here is kind of a little pie chart to show you roughly how much time relatively is spent on each level here. This is interphase. So the overwhelming majority of the cell cycle is spent in interphase. And that's when the cell grows, when the DNA is copied. It's copied as chromatin, and it's prepping for being divided. If nothing was copied, you'd wind up with a smaller and less complex cell every time it divided. But everything replicates, and it gets bigger, and then it cuts in two, and then that keeps happening and happening over and over. Okay. The DNA replication is kind of similar to transcription when we talked about protein synthesis. But the difference is instead of the DNA opening up temporarily and then closing back up, it opens up and it splits and a new copy is made. So one string of DNA opens and it makes two new strings of DNA that are identical copies of themselves, okay? So this requires enzymes. Again, a special enzyme called a helicase, right here, is what unzips the DNA this time, okay? Another enzyme, which is called DNA polymerase, super fancy, it helps get the nucleotides lined up in the right spot, okay? And it lines them up on the unzipped side. Okay, so remember, here's one unzipped side, and here's the other unzipped side, and so there's new nucleotides coming in on this inside unzipped part. Okay, and then two identical pieces of DNA are eventually produced. So we've got this strand that eventually closes up, and you've got two of them that are a copy of this initial single strand. Okay. So that's happening in interphase. That's known as DNA replication. Now, mitosis, it's four separate steps that really preps the cell and gets things lined up. We'll talk about those 4 steps. And then that last step is cytokinesis, okay? After interphase, after the copying and the growing, we have prophase. During prophase, the DNA condenses into those chromosomes. It's where they look like the X's and you can see them. There's a lot of stuff here. You should probably pause the video after I get all of the parts up here because there's a lot going on. There's something known as sister chromatids that are connected, and we'll talk about that a little bit more later. And these spindle fibers attach. These things up here are the spindle fibers. That's what's going to attach to the chromosomes and actually pull them apart eventually, okay? And another thing that happens in prophase that I didn't say, the nucleus is surrounded by a membrane. It starts breaking down because those chromosomes have to come out of the nucleus, okay? All right, then we go to metaphase. The chromosomes line up along the equator of the cell. You get this nice sharp line of chromosomes in the middle, and you can see those spindle fibers are attached to them, okay? These guys on the ends here, they're called centrioles, and they're like little anchors, and they hang on to those spindle fibers, okay? Then we move on to anaphase. Those spindle fibers are going to start shortening. The anchors stay put. Spindle fibers start shortening, and it separates those chromatids, okay? Those sister chromatids from up here get pulled apart, and so each side of the cell now is going to have a chromatid, okay? And then finally, telophase is when the daughter chromosomes begin to uncoil, nuclear membrane starts to reform. We don't have two new cells yet, but we're almost there. It almost looks like this figure eight. Things are starting to reform. The DNA is uncondensing, and this shape of two new cells is coming. I'll come back to this in a second. And then finally, we wind up with cytokinesis. And cytokinesis is that last step that pinches off the last little bit of the connection between the cells right there. And you wind up with two identical daughter cells, okay? So one cell grows, copies, makes two cells. And then they start the process all over again. Interphase will begin, and then those daughter cells will grow, copy, and then become four cells, okay? So this is a process that replaces dead cells. This is a process that allows us to grow and replace things like skin cells, but it has to be controlled. If it goes unchecked, imagine if our cells just kept dividing. We don't grow forever, okay? We don't become ginormous, people and blobs. And so eventually this does slow down. If something goes wrong with this process, this could actually develop into cancer. Cancer is basically cells that are dividing uncontrollably, okay? We'll talk about that more later. All right, let's go back to this slide here. Just so you can see some additional pictures of those steps, okay? Again, interphase was the copying and the growing. And then mitosis itself is these, here's my pen. It's these four steps in the middle. If you remember the saying, I-P mat, I don't really know what that means, P-mat. But anyway, if you say that to yourself, you can keep these in order, okay? So prophase, the chromosomes are formed, they're out of the nucleus. Metaphase, they're right along the equator there. Anaphase, they get separated. Telophase, two new cells are almost forming. And then finally, we've got cytokinesis, where it pinches off and you get two brand new cells, okay? That's it, mitosis.